the results of our investigations as to how share would be acceptable to the patients and the researchers. So this is one of the underpinning publications before we actually really got going on the register. So, so in terms of recruiting to studies, well, recruiting into share, sorry, we've used a lot of public engagement to do this, including social media, and advertising, and also face-to-face recruitment in outpatient areas, like the photograph at the bottom. Outpatient areas in hospitals have volunteers going around basically signing people up to this. That's actually been the biggest one. But obviously, we've been going for science festivals as well, and things, and any high density area like Freshers Week at university, the reception week at university, but also emails from employers to employees, just telling people about it and things like that. So face-to-face has been really important. Supermarkets, football matches, everything, and emails, and also letters. All the hospitals send out outpatient letters, and basically a leaflet, a consent leaflet goes out with the outpatient letters. And we've had advocacy. This is you won't know this, but this is a TV personality from morning television in Scotland, and in fact the UK. It's Lorraine Kelly. She's a famous hospital a famous television presenter, and this is from Edinburgh. This is two of the high-profile football managers. So this is the football managers from the two opposing teams within Edinburgh, Hearts and Hibs. We had to. This is probably one of the only times you would get these two guys in the same room. So that was great. That was all really publicity to drive registration to the register. And so now we've got over 280,000 people signed up. This is the front page to the share register, and this is a live tally of those signing up. And I think that was from yesterday. That's the tally from yesterday. This is just an old one. But we basically can follow through time with all the records. This is kind of an oldish slide, but this is just giving you some sort of idea of what we can actually we can see within the register. We can keep up-to-date records of what drugs people have been taking within the cohort. So you can see that within the register, 130,000 people have been taking cardiovascular system drugs, and you can see that across Scotland we've got signed up. We've got 19,500 with any sort of cancer registered with the thing. And of course, this is not data derived from people sign up. This is accumulating data that's updated all the time on the register. So, and this is just this age and gender profile. We generally, with most studies, we have an excess of females registered. Interestingly, as soon as you get to past the working age, that equivalence, that bias completely disappears, and you can see that more clearly here. Basically, it's young males just don't sign. Working age men are very difficult to reach for any of these sort of activities, and that's it's not just true for reaching this. It's also that if you actually looked at the number of the age and sex distribution of blood sampling by the National Health Service, you would get a picture exactly looking like this. So this is basically very reflective of the health service engaged population. It's not reflective of the entire population. And so for researchers, basically there is it's the same website where researchers can come in and they can actually start engaging with the team here, getting feasibilities, project applications, and all the information about how to apply for a project is all available on the website. Now, as I mentioned, we don't actually just give out lists of people who've got cancer, or people who've got any sort of diseases or anything like that. This is basically a recruitment portal. So if somebody has a project and they want to get some people, basically we basically generate lists of individuals and who might or might not have the disease. We don't say whether or not they're cases or controls to the investigators. That's blinded, and then basically the patient name and contact details is given in this secure online fashion. These individuals are then they can be contacted by the investigators, but then basically they have to tell us what happened with that patient. Did they take part in the study? Did they not take part in the study? Did they decline? Why did they decline? And then basically, as soon as that's finished, as soon as they are finished with that patient, that data disappears from the system. So it's a very carefully choreographed system where we only give people the individuals at the time that they need them. So we don't give people a list of 5,000 patients because they can't contact all 5,000 patients. We basically work with the study team to feed them the information in a timely fashion that their clinical team can actually reach out to them properly. So that's what we do. And an example of how share actually is useful is here. This is a study that that we actually did years and years ago. It was called TRACE-RA, and it was looking at statins for rheumatoid arthritis, and basically it failed to recruit. It took 5 years. It was a study that used the rheumatology network across the whole of the UK, and it ran out of steam after 5 years of trying to recruit. Again, a lot of it was, again, the inclusion criteria. Running them in an RA clinic was fairly, the density of suitable patients, even in our RA clinic, was very, very low. But if we did this within SHARE, we could actually do this in 2 to 5 weeks to identify the patients and reach out to them. So just using the fact that they've got a particular syndrome and what drugs they've been taking. So I mean, this is how SHARE is advantageous. So we have over 280,000 volunteers signed up, but we've also got over 120,000 spare blood samples collected. And we've also had, I think it's nearly 200 studies approved now and going through there. The other thing is that about letting people know about studies. It's not just us actively reaching out to them. We also give them a way to see what studies we're doing and a way to come straight in to the trials. And this is basically how we manage it, but this is basically, we can actually set in the study section in our trial portal, we actually can do triage of patients and let them go directly into clinical trials through the SHARE website. So that basically drives more SHARE registration, and it gives an alternative direct entry mode for SHARE patients into clinical trials. Now, move on to the spare blood aspect. So you can see here, this looks awfully like that graph, doesn't it, of the who signed up to SHARE. But this is in fact the whole blood test, the whole blood counts in hematology in Tayside over the years 2008 to 2011. And you can see that, that the females are being highly tested all the way from late teens all the way through. Whereas the men are basically don't go to the doctors, they don't get blood tests when they're young until they start to get a bit older again. And again, the whole thing changes at end of work age. So that's whole population data, that's not consented data, that is of the whole population. And what we actually ascertained from this, and this is what got us going, was that if we had everybody consented tomorrow, in 3 years, we could have blood for nearly 70% of the entire population just by storing the routinely collected blood. So that was a major impetus, the pilot data, to make us do this spare blood project. And this is one of the adverts that we had for this So how does that work? Now it's a real problem because most busy laboratories in Scotland have got tens of thousands of samples going through their laboratories every day, and to bioresource all of those and to find those who've got consented bloods manually is absolutely impossible. So we had to develop a completely in silico mechanism, and you saw there in that video, you saw the use of conveyor belts and robots. So basically the whole process is completely automated. Basically we flag the NHS system for the individuals who are in share, and then automatically the NHS blood sciences systems put those samples aside for the bioresource labs. And then importantly, we say, oh, okay, we've got a sample for that patient. Please do not give us any more blood, because many patients, especially when they're in hospital, might have over a period of a week 20 samples or something like that. So you can end up with many samples for patients. Now, obviously, in some cases that might be useful, and I should be able to take you through how we can use that particular component. But for genetics, for instance, we just want one sample from that patient, and obviously don't want to fill the freezers with too many duplicate samples. And that is totally electronic, and we have basically developed a safe and secure and high performance infrastructure for analyzing any of the data, for managing the data from these samples and linking into the clinical data set for both approved researchers. But this is basically a data set that could also be an absolute gold mine for the NHS for clinical decision-making supports. And I think Alex Donny in the next talk is going to talk more about that particular component of the thing. Now, we have been collecting longitudinal samples from the share, not just the genetic samples. So, for as a pilot, we basically have said, let's everybody who is in Tayside who's diabetic, let's just store all of it and make plasma from all of the samples, and then we can actually start to answer some questions that you couldn't answer in any other way. And this is just an example of a study that we did looking at people who'd had myopathy on a particular on statin lipid-lowering drugs. And here we basically had a series of repeated plasmas with some individuals where we had blood before the prescription of a statin, but then we also had blood and plasma stored after the the clinical event of myopathy. So we basically could do some lipidomics here, and this is just the results of, I think, and what was interesting was that the only replication we could do of this study was actually on studies where they had taken, the only sample they had was after the myalgia. And this is, and what we found was that the results before predicting the myalgia mirrored those after the myalgia, and it was all to do with arachidonic acid depletion and icosanoid generation. So it was really a very interesting study. And this is just some examples of patients. So that these two graphs are two patients basically. And each blue bar is a blood sample. The y-axis is the volume within that blood sample. And the top sample is a diabetic patient who, during the summer of 2015, had a very messy hospital history, many samples. And the one below that, which is fuzzy and pretty terrible, but is a type 1 diabetic patient who comes for a screening visit every month. So for this patient, we've got monthly serums for you. So these are invaluable samples for doing longitudinal analysis on biomarkers within patients. Now, what have we been doing for genomics here? Well, we've been using the Illumina Global Screening Array. And this has been a very cost-effective tool within our group, and because it's not just a GWAS tool, it's a tool that's very useful for clinical research variants, and very importantly, pharmacogenetics. It's got most of the pharmacogenomic variants that are known on there. In fact, it's got everything pretty much on there for determining the 140 genetic tests that are currently labeled by the FDA. And this is what we've decided, the very first example to really work with SHARE was to really try and implement the clopidogrel pharmacogenetics, which is 2C19 metabolism, where we basically have 30% of the population which barely metabolizes clopidogrel. And this is data from Alex Donny, who's going to be talking in a minute. I'll just go through it quickly. Basically, he showed that we can see in stroke patients that they're essentially untreated, and there's lots of papers of this has been known for many years, but we still don't use it in the clinic. And it's way past time that this should be used in the clinic. And so that's one of the things that we're trying to work to provide as a benefit from the shared data set as an exemplar. So obviously this is a population you've already heard about, the Estonian biobank. This is another contribution to what we need is these large-scale population biobanks. So we started in the early 90s with diabetes projects in the low thousands. But that's just been ramping out now. We know we need to study so many people, especially when you've got so many complex clinical and health stories that you need to ask the genetic questions about. So this is just an ongoing activity where we really need to go forward. And this is an example of what we produced from SHARE, which was we found a novel immune gene variant, which is basically associated with muscle pain and statin intolerance. And this was shown both in SHARE and in other local bioresources, but also in the world's largest statin clinical trial, Jupiter. So this is an incredible immune finding where basically people are more likely to find their statins basically unacceptable to them, and they stop taking them. We've also been, so the other thing that we want to use within SHARE is to start using genomic risk scores. And this is a really good example, where we've got for heart disease, we now know hundreds of genes for which each have got tiny little effects on heart disease. But when you add them up, they start to become very informative. And in this case, the Canadian group identified that if you took the top 2% of polygenic risk they've got a 6-fold risk of having heart disease before the age of 40. And that compares with the same amount of risk that you actually get with the most common monogenic form of CAD, which is familial hypercholesterolemia. So the actual sort of population burden of risk is much higher with these polygenic risk scores than it is for the monogenic diseases. So this is something this is one way to really find people who are at very high risk of CAD early, but have no other risk factors. And the people that are having heart attacks here, you wouldn't spot them using any other methodology, cholesterols or blood pressures or other risk factors like that. So this is the way that people are starting to think that polygenic risk scores are going to be done. The other aspect of SHARE is combining the consent to contact and the genotyping, and being able to use the genotyping. This is where we can actually design recruit by genotype clinical trials within the SHARE population. This is an example by Laura McCreight and Ewan Pearson, where they were basically looking at gastrointestinal intolerance to metformin. Now we believe that there's an interaction with antacids such as omeprazole and also the genotype of a metformin transporter, which and that's basically with this. So we basically managed to recruit individuals with null mutations in the transporter for metformin to do this. We've also run recruit by genotype for the statin intolerance gene, and we've also been able to provide individuals with high risk for Alzheimer's, genetic high risk for Alzheimer's, i.e. the ApoE4 genotype. We've been able to enrich those for companies wanting to trial new anti-dementia therapies. So, and of course, ApoE4 has got a very low frequency, so we managed to up the screen of efficacy of getting the people into those trials really massively within using the SHARE procedure. So hopefully SHARE will be able to produce, will be able to contribute massively to the oncoming genomics and drug precision medicine in the future. And this is basically the team here that is Team SHARE who've been involved in getting this going. And of course, you can't really do something like this in this post-digital age without having an app. And of course, we have an app. And this is a way people can sign up. It's also a way people can actually see what's going on within SHARE. It also allows individuals to provide us with additional data that we can't actually get, that might not be available through the health record. So we actually have it integrated into the Android Health, the Apple Health, where you can actually give consent for any of the health data that's on your phone, or you can manually input any data in there into that, so including caloric intake, height, weight, smoking status, and things like that. So this is something we've got going fairly recently, but we think it's something that will be very useful in the future. And I would like to finish there and thank everybody very much.
Thank you, Colin. Very interesting presentation. So do I understand correctly, it's more for Scotland and not the whole UK?
Yeah, no, this is not for the whole UK. It is a Scottish Health Research Register. Yeah.
Okay.
So yeah, the NHS is actually is a devolved process. So basically the Scottish government is in charge of health. So the NHS Scotland is a completely different entity to the NHS UK, which isn't helpful, I've got to say.
Yeah.
but actually, you know, in terms of, because of the scale, you know, some of the linkage aspects and things like that have been easier in Scotland for historical reasons. So a lot of this, you know, we're actually working actively with different areas in England to do similar things. You know, obviously a lot of people have been in touch with us from England. Can we do this? Can we do this? And we've got one franchised register which is called Discover Northwest London, and they basically franchised the platform and the whole procedure.
Okay.
And yeah, so we're looking at how this can work with what's called the Accelerated Detection of Disease program. So the UK government wants to genotype 5 million people, and at the moment, Share will be the platform in Scotland, apparently, hopefully, but it's not been agreed yet totally. So, but obviously we're trying to say in England that this would be a cost-effective way of doing it, because to actually go out and physically recruit 5 million people and take their blood actively is going to cost an absolute fortune.
Yeah.
Anyway, yeah. So that's but
it looks very, like, from a point of researcher, it looks very easy to use, more better than classical methods.
Well, I mean, the fact that the blood just keeps coming in is just, you know, there's no, you know, it's the amount of infrastructure required to get it running is so minimal. It's great, and the fact that you can get longitudinal sampling is quite unique as well. It's bizarre because it's kind of the other way, you know, it really coming in from the patient engagement side, rather than the biobank is almost like the back end of it. That's not the front end of this. Although when we talk to patients about it, it's actually the biobank, it's the spare blood component that makes them excited, which is quite, you know, actually, because it's actually harder to explain, and so in fact people are more suspicious of some of the other aspects of health research, especially clinical trials. But it's not just clinical trials that we're doing. There's lots of questionnaire work, lots of quantity, lots of experimental medicine, you know, lots of focus work, and actually one of the biggest groups of researchers that have come to us are patient involvement. So basically, people come to Share to get people to help them design their studies.
Okay.
It's not.
Share clients are more like scientific institutions than industry, or it's both?
Sorry?
Like Share clients are industry or scientific?
Both.
Both. Okay.
Both. Yeah. But I mean, a lot of the academics that come to us are actually looking not for patients to give pills to or to take their blood. They're coming to they want to do lots of questionnaire work, lots of focus group work as well. So we're basically enabling the whole range of health research, you know, from focus groups, patient involvement, you know, in designing new studies, to hardcore commercial clinical trials, recruit by genotype. You know, the diversity of the health research that goes on within the register is just immense.
Yeah, and it's with the app and everything, it's pretty user friendly. Yeah. I imagine, yeah.
And on that, you can go and they can see what studies are ongoing and do they want to take part in them. They can volunteer for the studies through the app.
And maybe you have also a solution how to just like about the microbiome, how to record like what people eat and how often and this kind of. field?
But I mean, yeah, I mean, so the app has got the functionality of taking anything from the health app, and the health app does, you know, you can. So one way of doing it is to actually point people to a particular program that actually collects dietary intake, and then that data will automatically populate the shared database. So say you have a study which says you want to track somebody's caloric intake and nutrition load.
Yeah.
You can just point them to something like an app like MyFitnessPal or something like that. If you use that, and you let Share have the data, then that will be automatic.
Oh, okay.
Yeah. So basically, it uses the integrated functionality of the health apps, and of course you've got control using the health apps as to which data you share and which data you don't share.
Okay, and is it like patients don't get like their individualized results for now? Yes.
No, not for now. We are looking at ways of doing that, and that's something that we would like to do. But we're still working on how to do that, you know, in terms of genomic feedback and things like that. But it's too expensive for us. We haven't got that would, you know, there's a lot of issues to deal with before we can actually achieve that. But we'd love to do it.
And another question is like about this bias between female and male participation. Do you also see like a bias in minorities and like other ethnical groups that are living in UK or Scotland?
In Scotland, that data that we're actually just interrogating that data at the moment. I don't really have a good handle on that at the moment. Based on the PCAs of stuff that we've done with the genotyping, I would say that we're getting a fair old mix.
Okay.
Because in particular we're comparing the shared genomic data to Indian data at the moment, and so we can see that we're getting a good representation of South Asians, which are the main immigrant population in Scotland. So we're getting a fair old representation there as well.
Okay. And from me also, the last question is: do you think this biohacker and longevity movement will also improve participation of men? Like
yeah, I mean, obviously, men are as they get more engaged in, I say, the bias is totally reflective of how engaged a man is in the health process. Exactly. Yeah. So any of these movements where people are more aware and thinking more about their health and how they can be proactive about it will obviously draw them into something like this.
Okay, thank you very much. That's very interesting and very nice idea to, like, I can see it can be very useful.
Okay, thank you very much, and very sorry I was late today.
I think it's okay. It's fine. We needed a little break anyway.
Okay.
So if no one else has a question, I think no. Then yeah, we can say bye for now and.